My website is cucurbitbreeding.com. It's partially maintained by ads, but I have to pay about 2/3 of the cost. But if you want to find out more, you can go to the website and click on Wehner, and you'll see resume and other things there. I was originally assigned to work on Old World cucurbits, so cucumber, cantaloupe, watermelon, and gourd. The New World cucurbits are squash and pumpkin. So I work on all the Old World cucurbits, and then the funding started to go down for cucumber and for watermelon, and all of a sudden there were these opportunities in stevia, and I just happened to be working on monk fruit. Anybody heard of monk fruit? A few? Monk fruit is a cucurbit. It's like a melon, but you harvest the fruit and use the compounds in them for the moghricides that are low-calorie, all-natural sweeteners. But it's really hard to work with monk fruit, so I started working on it, and then I stopped working on it. And then I got an opportunity on stevia, which is also a low-calorie, all-natural sweetener. So you'll notice on my website for cucurbits, there's a stevia area. So I put today's talk on the website so you can read about it if you want. I also made some slides for you. I usually only use PowerPoint for pictures. I don't usually use PowerPoint for talks because when I teach, I don't know if any of you teach classes, but studies that have been done where people teach with PowerPoint versus chalkboard and then switch halfway through the class so that each lab gets the other treatment. The chalkboard class always does better than the PowerPoint class. Because if you sit and write while you listen, you learn more than if you sit and look at PowerPoint slides. So this is my talk, sort of assembled on the web, so you can look at it later if you want to know. But so people are looking for ways to get around eating sugar, because the only thing that nutritionists seem to be able to identify as bad, I mean, they're arguing about fats and carbohydrates and proteins. They're not quite sure. They change their mind all the time. Should you have eggs, or maybe just egg whites, or whatever. So the argument is going on. But the only thing they agree on is you shouldn't have sugar. If you had no sugar, you'd be great, right? There's no reason ever to have sugar. And people are now starting to pull it out of some of these foods that we've been buying, so there's not quite so much sugar sometimes in those. But they agree that you should not have sugar, and they're trying to figure out the alternative. There are three. There's sorbitol, if you're looking for natural, low-calorie sweeteners, right? There's sorbitol, that causes problems with the runs, the runs. It's good for old folks like me to eat prune plums, for example. They're full of sugar alcohols like sorbitol. So that doesn't work very well. And there's monk fruit that I just told you about. Lohan guo is the name in China. Guo means cucurbit, so huanggua, cucumber, hamigua, cantaloupe. Anyway, Lohan guo is the monk fruit. And there's stevia. So there's three. And unfortunately, I know legumes, that's what I did for my graduate work, and I know cucurbits, but I don't know sunflowers. This is an aster. Stevia is an Asteraceae. And it's so I worked on self-pollinated legumes, then I worked on cross-pollinated cucurbits, and now I'm working on clonally propagated asters that are perennials. It was like you couldn't get any more different, right? If you wanted to do plant breeding, it's completely different from what I know. So I've been learning. I should have learned this in plant breeding class, but I ignored it because I was interested in legumes. But I should have learned how to do clonal breeding, so I've been learning that. And the crop is basically, since only 2008, the FDA approved stevia in America in 2008. So it's very new. The Guarani people used it in Paraguay, which is the center of diversity. So if you want to get stevia, you should travel to Paraguay, the most probably primitive country in South America, landlocked, sort of Andean jungle, a little bit of the plains in there for Argentina, on the Argentina side. But anyway, the Guarani people in Paraguay use this as a tea, and now we're starting to use it in the US. And it turns out, you all know that, how many of you know the #1 crop in North Carolina? Anybody know the #1 crop? What is it? Tobacco. And we're trying to figure out how to get away from tobacco, and growers are trying to switch to other crops. And we've been switching to other crops really for my entire career. We've switched to strawberries, cucumbers, sweet potatoes, I mean, everything you can think of. But we still have, of course, the problem of most of those only have small acreage requirement. So Stevia uses all of your tobacco facilities. You can use your greenhouses, the float greenhouses. You can use your transplanters. You can use your tobacco drying barns. Pretty much all the methods are the same. None of the tobacco diseases affect stevia, so you can grow stevia and get away from all those horrible diseases on your farm that you've been fighting on tobacco. The only thing you need is a harvester. You have to have a harvester. And you can buy a snap bean harvester. Are we defending something? Something's attacking us here. Huh? Windows Defender. So you just need a snap bean harvester. And you set the snap bean harvester a little bit too aggressive, and it rips all the leaves off the stevia. It's really great. New crop, replacing or going along with tobacco. And the problem is you have to have a different formulation with every product. Sugar is great. You can put it into solution, you can put it into cakes and cookies, you can put it into colas, lemon lime. You don't have to worry about it. Just use the same sucrose. All the other things, you have to have one formulation for cola, another formulation for lemon lime, another one for baked goods, another one for use as whole leaves in tea. So everything, and there's also table sweeteners. Where's my table sweetener? So it's probably dumped out in my pocket. You know, so you can get these table sweeteners. This is from your PHHI. And people who eat stevia, when they try it, half of the population of humans doesn't like stevia. Half of you don't like stevia. How many of you don't like stevia? See, you've even tried it and found out that you didn't like it. Now here's the problem. We're developing new stevia that will, we hope, taste good to you. And you already think you know what stevia tastes like, so it's going to be difficult now to convince you to try it again. But we're coming out with some new varieties that will taste, we hope, will taste better. So here's the basics of stevia. It's an herbaceous perennial, so you could grow it. Some people are getting 5 years. They're growing it for 5 years. Probably 3 years is what you would grow it. If you're in the mountains, or if you're in the north, or the south, meaning southern hemisphere, you might only go 1 year. You can treat it like an annual. A lot of growers in China are treating it like an annual. They put it out at huge densities, and then they harvest it and start over. Okay, so you can treat it like an annual, but it's as I said, it's a sunflower family, Asteraceae from Paraguay. It has been collected. There are expeditions that have gone out and collected stevia accessions as well as different species. There are actually lots of species. And so India has a large collection. They made it in Mexico, Central America, South America. Big producers, producing countries have collections: China, Japan, Indonesia, and Turkey. And not coincidentally, I have 2 Turkish scientists working with me now on stevia. The problems with stevia are so numerous. You could, do you remember Jane Goodall when she went to study chimpanzees? She went to study chimpanzees for her PhD. It was supposed to be just for a few years, right? She went off to someplace in Africa. Where was it? Gambia. reserve or something. So her advisor, when she left, said, you know, this is going to be your entire career, right? And it was. She never really finished with chimpanzee studies. And the same thing is true of Stevia. This is going to be an entire career. There are so many problems to solve. Anything you want to work on hasn't been done, so it's a good research project for you. There's sporophytic self-incompatibility. You can't self-pollinate these things. There's germination problems. We have, last year, nothing germinated. But usually you get around 80% germination. The black seeds germinate, the brown seeds don't, okay? And I'll show you pictures later. There's a picture on the website if you want to look at it, but I'm not going to bother with it here. So the production methods, as I said, are just like tobacco. You can do all your tobacco stuff. You basically plant on bare soil, flat. You use transplants. We're on 30-inch spacing. We have 30-inch row spacing on our harvester, so the machines are all set for 30-inch rows, but you can go down to 15. And we go actually 6-inch between plants. You can go even narrower if you want, higher density. Greenhouse float beds. Do you all know what greenhouse float beds are? You actually put wood frames in your greenhouse, and you put plastic liners over the wood frames and fill them with water. And then you get those styrofoam trays and float the trays in the water. So Stevia loves water. If you see a puddle of water on your black plastic mulch in the field, you'll see seeds germinating out of the water in the field. You want to see the picture? Or I got a bunch of pictures that I'll show you later, but I can do this if you want to see this. Okay, here's a float greenhouse. So this is, you see Veronica is standing in the row in the middle, and then there's a piece of wood here, a 2x4, with black plastic over it, and these are all floating in water. You can see the black plastic right here. That's the end of the bed. It goes all the way up to where I'm standing. And these are all floating in water. The water's not very deep. The roots are just a few inches down in the water. And you have a pump that circulates the water, and you put fertilizer in the water. It's just like doing hydroponics, right? Except these are for seedlings instead of plants for harvest. And we put chemicals in the water like TerraMaster that prune the roots at the bottom of the flat and also kill off the pythium. But you can also do this organically if you like. So basically what you're doing is you're planting seeds in these float trays and preparing the transplants for field use. It would be better if you could plant the seeds directly in the field, but they don't germinate very well, and we've had a lot of trouble with that, so we've had to use transplants. It would be better if you could use transplants produced from seeds, but the seeds are all different. It's like planting apple seeds. You know, Johnny Appleseed went through the whole eastern US planting apple seeds, but they were seeds from the varieties that he liked. They weren't the varieties that he liked. They were seeds from the varieties that he liked. Half-sib families. Every tree that came up that Johnny Appleseed planted was terrible, right? They're all terrible. They're what we refer to as cider apples. You know what that means? You grind them up and you make alcohol out of them, right? So a few of them were good, a few of the trees were good, and those were clonally propagated, and that made these, varieties that we use now. So Red Delicious seeds do not make Red Delicious apples, right? You can't use the seeds. So same thing with Stevia. A really great variety of Stevia is clonal. If you plant the seeds, it's not that great variety. It's a piece of junk, right? And it's variable. So that's what we're trying to do, is how to plant the seeds. We want to know if we can direct plant or at least plant in flats. Unfortunately, we have to clonally propagate in flats and transplant. It's like the worst case scenario. And it would be worth it if it was watermelon, but it's Stevia, which is just sold for dry leaves, so it's not very, let's see how I can say this. You got to do it cheaply in America with lots of machines if you want to make money. Okay, what you want in Stevia is the leaves, because there's a compound in the leaves called stevioside, which is related to gibberellic acid. It's like a hormone. It's one of the plant hormones. So steviosides seem to be on their way to becoming gibberellins in the plant, but somehow they get stopped partway and they become steviosides. And rhodioxide A is the one that people, so originally they sold Steviacide, but it had bitter aftertaste, so they started selling rhodioxide A because it didn't have quite so much of the bitter aftertaste. And now we're moving on to other rhodioxides. There are many of them, and they seem to have better and better taste with less and less of the bitter or sour aftertaste. So we're moving towards better steviacides for better flavor, but we're still working on that part. And they're about 300 times sweeter than sugar, so you don't need much. How much time do I have, Penny? 11:30? With questions at 11:15? Let me shortcut this to the pretty pictures. So just to finish the website, we're working on the breeding part. We also have other parts. If you go to the top, you'll notice that we have NC State. We have a group at NC State. Dave Hsu is doing pathology. Roger Batts is doing herbicides. And don't tell him that I forgot his name when you talk to him. But when you get old, you start to forget things. Gary Bolin is doing ag economics. So he's telling the growers the economics of stevia, which right now it's not a crop. Stevia is not a crop. Don't grow it. It's not there yet. He's working on the economics. We're working on marketing. We're trying to figure out if there's a processing plant we can build in the US. Right now, there's no processing plant. You can buy plants and you can sell leaves. So if you can line up a person to buy your crop, then go ahead and do it. But so we have an NC State group. We have a USDA grant that's organized out of Michigan State. So we have the USDA group with Ryan Warner, not to be confused with Todd Wehner. And we have a Pepsi group. So my funding is mostly from Pepsi. So I am actually not telling you on purpose. I'm purposely not telling you anything about the Pepsi side because they would come and drag me out of here. Let's switch over to these photographs. Does this thing change slides? Good. Okay, my technician is Emily Silverman. She's also working on her PhD in stevia breeding. So here's some pictures. This is the way stevia looks. That's a single plant. It's a perennial shrub, narrow strapped leaves, and it is vegetative for most of the summer. Then it turns reproductive sometime around September. If you want to get the highest yield, you should wait until just before flowering, which is all over the place when you look at these things. It starts to flower sometimes at transplanting, sometimes they go all the way into the winter, sometimes they never flower. So it's just a real mess to work with. But you can take these leaves, you can make cuttings. Every node will make a plant, so you can make nodal cuttings, leave a couple of leaves and a node, and you'll be ready to go if you want to make more of these. Here are some of the products you can make with steviacides. Most of these are actually made with rhodioxide A. If you look on there, it usually tells you what the sweetener is. Excuse me. Okay, so this is the structure of can we get rid of this sidebar here? What is it? The bottom? Down?
Put in presentation mode.
Oh, I'm in the wrong thing. Oh, yeah, I forgot to click that thing. That's right. I knew that. I was just testing, make sure you guys knew. It's hard to believe, but I used to teach presentation skills for graduate students. Don't tell my boss. HS 601. Okay, so anyway, these are the steviacides. So this has 3 sugars stuck onto these rings. So your tongue tastes the sweet, but the ring keeps it from being digested. No cavities, no diabetes, no calories, which really would help me. Here is rhodioxide A. It has 4 sugars. Here is rhodioxide M, as in Mike. It has 6 sugars. So M tastes really sweet and hardly at all any bitterness. So if you, here's the, this is a taste diagram. You notice here's the leaf, bitter. Here's stevia side, not quite so bitter. Here's Reb A, even less bitter, right? And still they're all sweet, but you notice Reb A is the most sweet. And then here's the pattern for Reb, this is M. Green has M with Reb D added. So you notice if you add Reb D, you really get rid of the bitter versus Reb M by itself, okay? You knock down, not a whole lot, it comes down some. It's not a huge amount, but it takes bitterness down some. You notice it still has some bitterness on there that we need to work on, but they're very sweet, okay? There are varieties available. You can get the Maritas and Irate and Kannapolis from Stevia store in Paraguay. The seeds don't germinate when they arrive. You might get one out of a whole flat that comes up and gives you a plant. And then you have to make more of those yourself. You can also go to garden seed companies. We spend thousands of dollars just buying seeds for trials each year. And also you can get heirlooms. If you go to Seed Savers, you can get stevia from them. I'm not sure if Seed Savers is selling stevia this year. They seem to be out of it. I don't know. Now, in the greenhouse, we make pollinations using these net cages, which I'm sure Massimo recognizes from onion breeding because that's where we got it. We do a little onion breeding in the greenhouse using stevia. And these are, yes, flies inside of plastic cages, pollinating away happily and then dying. There are fly carcasses littering the whole greenhouse floor. So we have a shop vac every morning. We take care of it, and you can see the flowers. You can see the flowers here. There's about 6 flowers in a cluster. And flies will do it really nicely for you. It's hard to do it by hand. We've been successful, but it's pretty difficult. Here are the seed and flower parts at harvest. So these are being harvested and bagged. These come from a biparental cross in a large cage. And then we run them through a seed cleaner. And I need some way to take the glooms off of these seeds. I was looking at some of these de-hullers to see if they would work, but I haven't found a solution yet. But the objective is to get black seeds, not brown, and take the pappus off of the seeds so it can be planted without problems of dropping through the vacuum seeder clean. We need them to go through cleanly. So, and then we plant these in the greenhouse. We maintain mother plants, and we take the mother plants and propagate those. So you see we have growth chambers for propagation. And we put them in the greenhouse. So these are cuttings being propagated, and we use mist overhead to get the cuttings to establish. And then the seeds, what we do is we take the seeds and we put them into float beds. So this is sort of like the tobacco float greenhouse, but this is, of course, a hydroponic float. And you put the flats in there, and you can see after a couple of weeks, you've got little plants germinating. And this is actually really good germination on this one. But a lot of times you plant and you see like one plant in the flat. And then after a while, they're in the float bed and they're growing nicely. And there's nothing wrong with your vision. We actually have different color lights. So we put white lights on when we're just doing general growth. We put blue lights for vegetative and red lights when we want them to flower. So these are $35,000 worth of LEDs over the top of this greenhouse section here. And this is the grower greenhouse, except of course it's not a grower, it's our greenhouse. We now have greenhouses at Whiteville, Clinton, and Oxford. So we have actually barns and greenhouses and fields at 3 locations. So we're doing 3 locations of testing. And you see here, that's the center aisle. And then these are floating. And there's overhead mist on one half and no overhead mist on the other half. You don't see it in this picture because it hasn't been installed yet. But The mist side is for cuttings, and the no mist side is for seeds. So we run roughly, the program is roughly double. Seeds and cuttings are about the same size in the program. So this is the effect of using regular flats versus float trays. They're the same age, same variety, same location. So that's why we use float trays. It gives you a huge speed gain relative to regular flats. And speed breeding is where we're trying to go with Stevia. Speed breeding is the use of multiple techniques to try to get as many generations per year as possible. Right now we're getting 1 generation per year. So that's 2015, 16, 17, 18. I'm a 4-year breeder now. So yeah, we have a long way to go. Then we put them into cold frames in the field. Then we print steaks. And this steak printer spends most of its life being broken, but when it's working, it prints about 20,000 steaks for us. And then we try to thin the flats, so we only get 1 plant per drop on the transplanter. Then we drop them into a lettuce. This is a lettuce transplanter. This is not our transplanter. This is the machine we bought. Ours looks like that. Does 2 rows. Everything for our system is set up on 2 rows. So every plot is 2 rows, 30-inch between rows. So 2 30-inch rows or 60-inch width is 1 plot. And then we have 60-inch rows for the single plant selection. So these are 60-inch between row centers. So we have double 30s for the agronomic data and single 60s for the single plant data. So it's, unfortunately, we destroy all of the double 30-inch rows when we take data. So when we take agronomic data, so we have to have another set where we take single plant data for things like glycosides. So basically we're doing like a 30 plant plot versus a 6 plant plot. And now this has changed. We're now using bare soil transplanter instead of hand transplant on plastic. So this was 2015. So seems like every year we change as we learn, because as we're running the breeding program, we're also running the optimization. So as we work, we collect data, then we go back and look at the optimization and we realize, oh, we didn't do that very well, did we? And so we change, changing from black plastic to bare soil, for example, because the growers will use bare soil. So what it looks like is we have these double 30s for agronomic data, and right next to it we have single row 6 plant plots that we get the single plant data. So agronomic data, single plant data. Then the next study, agronomic, single plant, like that. So here's the way it looks now. Bare soil, double 30-inch rows, and some of those you can see we're having trouble with germination. That was actually a field day. It's amazing how many people attend Stevia field days. There's a lot of interest. So we got a suck-up plow. This actually throws the soil up on the sides of the transplants so that you get rid of the weeds and sort of straighten up the plants. This keeps the ones that are falling over, puts them back up again. Or either breaks them off. Either way, they're not leaning over anymore. And then we take data on single plants. We take data on things like disease. It's amazing how many diseases affect Stevia. And chilling, height, width, leaf size. And then we get glycosides. We harvest leaves and dry them for glycosides. And then we get cuttings, seeds, and then we dig the crowns in the winter. So the single plants give us a lot of stuff, right? We're getting single plant data plus cuttings, seeds, crowns. And then we get disease data also in our disease chamber. So this is just a cool, humid, dark chamber. And the diseases love it. And we also use the Phytotron for evaluating. We have things like Botrytis, Pythium, Septoria, tomato spotted wilt. You know how the ornamental guys hate us when we bring tomato spotted wilt into the greenhouse. It gets all over. I mean, ornamentals, are having huge problems with tomato spotted wilt. So we've made some selections. We have yellow leaf, that's a chlorophyll mutant. We have big leaf on your right. We think that the little leaf is the way to go, but we thought big leaf might be interesting for people who wanted like a tea, whole leaf tea. So we rate the leaf size 1 to 9, 1, 2, 3 small, 4, 5, 6 medium, 7, 8, 9 large. And then we harvest the plants, and the machine harvester actually weighs the plants, throws them out to the side, and then we put them in, well, we used to put them in paper bags. Now we use burlap because the plots are getting bigger. We're now moving from 10-foot to 30-foot plots in length, 2 rows and 6-inch spacing, so we're getting a lot more plants now. So we do harvest, weigh, fresh, then we put it in the tobacco barn and we dry it and we weigh and we get the dry weight. This is known as how many people know what this is? It's a leaf grinder. I know you think this is a hardware store paint shaker, but actually it's a leaf grinder. A Red Devil paint shaker. I highly recommend these. They're cheap and they don't break. You can shake the living daylights out of these tubes and they'll grind really well. There's a couple of ball bearings in the tube, right? So you put the tubes in there with the dry leaves and run the paint shaker, and those things come out in a few minutes just as fine as can be. The ag engineering shop built this for us, and the guy that was testing it for us accidentally threw a piece of wood through his head when he was doing it because he was making it out of wood for the prototype. And we said, look, let's just skip to the aluminum phase. No more wood. So here's our $150,000 forage harvester. It cuts, weighs, and throws out either left or right for the people to bag and dry. So that's Emily driving the haul drip. And if you adjust it right, it'll pick up 2 30-inch rows, and you can pretty much drive through the whole field in 1 day per location, instead of taking a whole week with poor graduate students and pruning shears going through the plots that way. We move from station to station for the suck-up plow, the transplanter, the harvester, using a trailer so we can take care of our stuff. Now, as I said, if you're commercial, you buy an oxbow green bean harvester. Oxbow. Set it a little bit too aggressive and it'll just take the leaves right off that stevia plant. And then you see the guy next is collecting the leaves in the hopper on the other tractor. So you got a self-propelled combine and a tractor. And then you're ready to dry and sell. We use tobacco barns. Here are the tobacco, we're still on paper bags in this picture, but we're at burlap bags now. And this is actually a grower's barn, but now we have our own barns, so we're starting to become a little more independent. But basically you load, there's the new barn. It's amazing how much they charge for those things. Now, in the field, we use cages for large-scale production. So these are 12-foot long cages with 2 double rows, so 4 rows in each. So you have line A, line B, line A, line B in the cage. So you're making biparental hybrids, but we're not using inbreds. They're biparental hybrids with half sibs. So we're making half sib times half sib crosses, which you can imagine what kind of mess that is. So we're trying to figure out ways to make inbreds, but as I said, it's self-incompatible, so you can't self it. Actually, you can. We've succeeded in making selfs on some. So we're trying to make inbreds, but I think it's going to be like onions. Like onions, you can go to what, S3 or something before the line dies out. So hybrid onions are really not inbred times inbred. They're S3. They've been self for 3 generations. So they're half, quarter, 7/8 homozygous, not completely homozygous. So so that's our field cages. That's inside the cage, and that's Bumblebees, we use bumblebees in the field cages. Flies in the greenhouse, bumblebees in the field. Those things are expensive, you know. Flies are a lot cheaper. And also, flies don't sting the hell out of you when you go inside the cage, so it's better. But we can't get bumblebees in those little cages that we use flies. We've tried flies in the field cages, and they work fine. So that's flowers. You see the flowers inside the cage getting pollinated. So what we're trying to do is get to the point where we can provide varieties for growers to use that will be adapted to North Carolina and also taste good. That's the objective. There are companies in the state that will provide you with plants and will buy your leaves. This is Hal Tea Garden on the right for U.S. Stevia, which is located in Laurinburg, down in southern part of the state. You know, the one that goes underwater whenever there's a hurricane. He's in the swamp area of Whiteville, Laurinburg. And we take data on overwintering. So basically, we rate them in February, March, about now, to see how they're doing for, you know, how well they made it through the winter. And then we're also, just a little aside, we're also running other studies. So we're using the Phytotron to look at things like photoperiod and temperature versus flowering. We have looking at trying to get clean seeds. So these are seed-borne diseases that we're studying. Trying to get tissue cultures, trying to make haploids from tissue culture or otherwise, so we can make instant inbreds. And before I take questions, I think I'm about right. So before I take questions, the group helping this year, Terry Williams, Danny Wisnowski, Danny Ryland, and Sonia Lombard are the ones who are working with us year-round. I have 2 visiting scientists from Turkey, as I said. Takshay Patel has finished his PhD and is now a vegetable breeder in Yolo, California, working for Volo. Volo and Yolo. And Sandy Wash does all of our bookkeeping for us because, you know, they moved all the bookkeepers out, and we don't have like what you guys have here in Kannapolis. So we have to have our own bookkeeper. So she costs me hundreds and she saves me thousands, as I say. So I know that was kind of fast. Sorry for the late start, but I'd be happy to take questions if there are any. Thank you.